Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper Shift 2. These are part B questions. This exam was conducted in July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR Net JRF Life Science being discussed, you can go to my channel Omni Bioentrance. In the playlist, select for CSIR Net JRF and there you will find. Now first one. Which one of the following adrenoreceptors decrease CAMP in the postsynaptic target after stimulation with norepinephrine? So it is alpha 2. Answer is option 2. In Drosophila stock, that is heterozygous null for a unique nuclear target gene was submated. Null means absence of a gene. Heterozygous null allele means one allele is present and one allele is absent. That is just like this. Here one allele is present, one allele is absent. One allele is present, one allele is absent. That is heterozygous null. For a unique nuclear target gene was submated. The target gene is essential for the development of Drosophila. The embryos from the cross were analyzed and the following results were obtained. PCR analysis of the genomic DNA isolated from the embryos showed that 25% of the embryos did not have target gene. If you see this figure, you can see that these are heterozygous null mutants. Upon crossing, you will get A minus, A minus, minus, minus. This minus minus will be the 25% which do not have the target gene. Then northern analysis of the RNA isolated from the above embryos showed the presence of transcript corresponding to the target gene. Now northern analysis showed RNA in all the offsprings. How come that RNA showed in the presence of this minus minus offspring also, that is the one with 25%? How come RNA came there? So this must be because of what is called as maternal effect. What is maternal effect? The offspring's phenotype is influenced by the gene expressed in the mother's genome. The offspring's phenotype is influenced by genes expressed in the mother's genome. That is maternal effect. And this is due to mother supplying the mRNA or proteins to the egg. So this must be the reason why all the offsprings showed the presence of transcript even though the DNA was absent. No lethality was observed in the progeny. Okay. Now, which of the following option best explains the above observation? Transcripts of the target gene are paternally contributed. No, it is maternally contributed. 2 is the answer. The transcripts are observed due to mitochondrial inheritance. It is not mitochondrial inheritance. If it was that case, it will be genes controlling the, it will be maternal inheritance, but here it is maternal effect, not maternal inheritance. Okay. The transcripts are being detected from the yeast that the larvae eat. That is also not correct. Then next one. Which one of the following statement regarding genetics of quantitative traits in plant is incorrect? Loci responsible for quantitative trait can show variation in their individual contribution to the trait. That's correct. Quantitative trait loci always have identical effects on a phenotypic trait in different environments. Not identical effects. They will not have identical effects. So that's incorrect. And that is your answer. They have asked for the incorrect statement. All the others are correct. Then next one. Mitotic cycline increases gradually through the G2 phase of the cell cycle, but the activity of mitotic CDK1 increases suddenly at the onset of M phase. This is because Now CDKs, they are activated in the cell cycle by cyclin binding, phosphorylation, removal of CDK inhibitors. So out of these three reasons what I have mentioned, something must be present in the options. We can see active CDK1 subunit is synthesized in the M phase. That's incorrect. Mitotic cyclin is sequestered in the cytosol. That is also incorrect. Activation of CDK1 requires post-translational modifications. Yes, that is through phosphorylation. Okay. Then the inhibitor of CDK1 is degraded in the M phase. It is not degraded, it is removed in the M phase. So 3 is the answer. Then the TI plasmid from Agrobacterium tumefaciens has genes for auxin, cytokinin and opine synthesis, while genes for opine catabolism and Vir genes lie outside the tDNA region. Okay. Which one of the following genes are involved in providing carbon source to Agrobacterium in their ecological niche? Now carbon source are provided by the opines, and opine synthesis and opine catabolism is there only in option 3. So option 3 is the answer. Then next one. A 160 kDa complex of 4 protein molecule consists of a dimer formed by a 25 kDa. So dimer, 25 kDa is a dimer. So it should be, dimer means 225 kDa will be present. So it will be 50. Connected by 2 disulfide bonds. The 3 other proteins of 10, 30, 70 kDa respectively. It is isolated and analyzed on an SDS page gel without DTT. That is, disulfide bonds are not broken. So this 25 kDa, it will be seen as a 50 kDa band, not as a 25 kDa band, because disulfide bonds are not broken. There is no DTT there. Which one of the following option would represent the SDS page profile? 4 bands corresponding to 10, 30, 50 and 70 kDa. Yes, that is the answer. Answer is option 1. Then which of the following is an example of parametric statistical test? It is unpaired t-test. All the others are non-parametric test. Then 2 students performed an ELISA to determine the amount of anti-spike antibody in the serum of COVID-19 patient. They used the same ELISA plates, the same reagents for coating, blocking, detection and the same ELISA reader. Both generated independent standard curves of absorption versus concentration using the same spike protein. Student A correctly reported the concentration of 100 microgram per ml, but student B reported 450 microgram per ml. So student B has got more concentration than student A, and student A is correct. It is already said in the question. So student B is wrong. Which one of the following could most likely explain the wrong result of student B? The ELISA plate was not washed properly between coating with antigen and blocking. If that was the case, then A also would have made mistake, but here only B is making the mistake. ELISA plate was not washed properly after addition of sample. That is also the same thing. If it was not washed, then both A and B would have made the same mistake. The slope of the standard curve generated by student B was lower than the optimal. Okay. Now this is the normal standard curve. Now if it was lower than this one, then the concentration would have been high. On this here you will have the absorbance marked, and on the y-axis, and concentration on the x-axis. Okay, so if it was lower than the optimal, then the concentration would be high. So 3 is the answer. The negative control showed very little absorbance. Negative control is usually indicate issues with background noise, etc. So not on concentration. Then next one. The tryptophan operon can be induced by the addition of indole propionic acid, which binds to the tryptophan repressor but does not change its conformation. Upon the addition of IPA, what will be the order of translation of the enzymes encoded in the operon? So first I will tell you what does this tell. Normal case, when tryptophan is present, what will happen in bacteria? Forget about IPA now. So when tryptophan is present, it binds to the repressor. The protein changes its conformation. The repressor and the tryptophan complex, it will bind to the operator, preventing RNA polymerase from binding and transcription of the operon. That is what is happening when tryptophan is present. When tryptophan is absent, what will happen is the repressor is inactive and it doesn't bind to the operator. This allows RNA polymerase to transcribe the tryptophan operon, turning it on. Now here you have IPA bound to the repressor and there is no change in conformation of the repressor. So it is equivalent to when tryptophan levels are low. Okay. There so R will be in the repressor will be inactive here. And it doesn't bind to the operator. This allows the RNA polymerase to translate the operon. Now they have asked how the sequence of the genes in the operon, in the structural genes in the operon will be. So it is EDCBA. EDCBA. Answer is option 2. Then next one. Which one of the following statements about bacterial operons is incorrect? Operons can encode multiple proteins with linked biological activity. Yes, multiple genes that are transcribed under a single promoter. That is operon. An operon expresses multiple proteins from a single mRNA. That is also correct. mRNA transcript of an operon has only one Shine-Dalgarno sequence. No. It has more than that is if you take the case of lac operon, it has multiple Shine-Dalgarno sequences. That means lac operon, it codes for 3 proteins and each with its own Shine-Dalgarno sequence. So incorrect statement is option 3. Operon expression is often tightly regulated. That is also correct. Then next one. Topoisomerase activity was measured in the term of change in the linking number of DNA in the presence of camptothecin, that is inhibitor of topoisomerase 1, or retoposide inhibitor of topoisomerase 2. Which of the following is the correct expected outcome? So in the presence of camptothecin, topoisomerase 1 will lead to change in the linking number by +1 +2. Okay. Now before that, topoisomerase 1 causes single-stranded breaks in the DNA and it changes the linking number by +1. Topoisomerase 2 causes double-strand break in the DNA and it changes the linking number by ±2. That much you remember. Now answer the questions. In the presence of camptothecin, camptothecin is an inhibitor of topoisomerase 1. So it will lead to a change in linking number? No. So 1 is not correct. Then in the presence of etoposide, etoposide is an inhibitor of topoisomerase 2, not of topoisomerase 1. Topoisomerase 1 will lead to change in the linking number by ±2. No, topoisomerase change the linking number by +1, not ±2. In the presence of camptothecin, camptothecin inhibitor of topoisomerase 1, topoisomerase 2 will lead to change in linking number by ±2. That is correct. In the presence of etoposide, topoisomerase 2 will be inhibited by etoposide. So it is not going to bring about any change in the linking number. So that is also not correct. Then A yeast strain has accumulated a mutation that make it grow slowly. Investigation revealed that ribosomal RNA levels have dropped drastically in this strain. Which RNA polymerase is likely to be mutated in this strain? So ribosomal RNA levels have dropped. So it means RNA polymerase 1 is the one that is drastically RNA polymerase 1 is the one that is needed for ribosomal RNA synthesis. So if ribosomal RNA levels are dropped drastically, then RNA polymerase 1 is mutated. Which of the following statements is incorrect? Phage mu is used to create insertion mutations. That is correct. Phage 1 P1 is the source of Cre-Lox-P recombination system. That is also correct. Phage 3 has single-stranded circular RNA genome, not RNA, it is DNA genome. So 3 is the incorrect statement. The other one is also correct. Next one. In which of the following proton motive force generated in mitochondrial electron transport is not used? Transport of ATP into cytosol from the mitochondria matrix. Electron transport is used. Transport of ADP from cytosol into mitochondrial matrix. There also it is used. Transport of phosphate ions from mitochondrial matrix. There also it is used. Transport of NADH from cytosol into mitochondrial matrix, it is not used. There for transport of NADH you use malate aspartate shuttle. Okay, so fourth is not correct. I mean that is the right answer. Then next one. Which of the following statements regarding the stereoisomers of D-glucose is incorrect? D-mannose is a C2 epimer of glucose. Now what are epimers? Epimers differ in the position of the OH group of one carbon atom. So this is D-glucose, this is mannose. If you see the carbon number 2, the position of OH is on the left hand side in mannose and it is right hand side in glucose. So they are epimers. Now allose and glucose, at carbon position 3 there is difference in the position of the OH group. For galactose it is at C4, carbon number 4, position of OH group is different from glucose. So 1, 2 and 3 are correct. They have asked for the incorrect statement. So naturally 4 is going to be incorrect. D-talose is a C5 epimer of glucose. That is not correct. It is a diastereoisomer of glucose. Now what is diastereoisomers? They have the same molecular formula and sequence of bonded atoms, but they are not mirror images or they are not superimposable. If you see the figure of D-talose here and D-glucose here, you can see that they are not superimposable, but they are not even mirror images. Okay. But they have the same molecular formula and sequence of bonded elements. Then next one. Which one of the following properties of grooves is a hallmark of the Z form of DNA? Z form of DNA doesn't have any major groove at all. So answer is 4, flat major groove. Greenhouse gas emissions are considered the primary driver of global warming through their influence on the radioactive forcing of the atmosphere. This radiative forcing occurs because long wave radiation emitted by the Earth's surface is absorbed and scattered. That itself is the answer. Then next one. The pH of water in Lonar Lake is found to be, they have given some values, taken once daily over 6 days. What would be the average pH of the water during this period? So average is, you have to add all these things, divide by the total number, that is 6. that you will get it as 10.4. But 10.4 is not there in any of the option. But if you round up each value, 10.56 rounded off to 10.6, 10.26 rounded to 10.3, 10.36 rounded to 10.4, 10.46 is rounded to 10.5. So what you got your answer, it is C, that is 10.36. That when rounded off will give you 10.4. Answer is option 3. So that's it with part B. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of CSIR being answered, you can go to my channel Omni Bio Entrance. Search for CSIR Net JRF in the playlist and there you will find. Thank you.